So next thing we're going to do is I'm going to give you a list of features that these cells have in common when I say these cells, prokaryotes and eukaryotes, or different types of cells. This pointer is giving me a hard time. So, okay, so you might have seen some of these before. We talked about DNA, maybe you heard about RNA, we talked about proteins. So each lecture is kind of going to build on it. We already talked about proteins last time. Now we're going to talk about some different kinds of proteins. And if you don't know some of these, I'm gonna go ahead and go over these today. So cell membrane, cytosol, ribosomes. Okay, so prokaryotes are the most ancient forms of life. So these are the oldest cell. They lack a nucleus. So if you look at this cell, you can see the material inside is just floating around. There's no nucleus. There's no center. And there's nothing around it either. And then eukaryotes, they have a cell with a nucleus and other membranous organelles. This is not the real size. This is not to scale. This is for comparison. Even just looking at this, you can see there's the size is a lot bigger and there's a lot more stuff inside. Those are called membranous organelles. So we're gonna look at this too today. So we're gonna see what's inside the cell. So one big difference, eukaryotes have a nucleus, prokaryotes have no nucleus. This chart is from your book, and sorry about the noise, it's somebody in the street. So this chart shows you some differences. So you have three big domains, and we talked about domain as being the first taxon. So you have bacteria, archaea, and eukarya. Bacteria and archaea both are prokaryotes. They are missing the nucleus. There's no nucleus. Pro means before, karyot means nucleus. This is before the nucleus. So there's no nucleus. There's no nucleus for archaea. Eukaryotes have nucleus. So there's nucleus present. And then we have something called membrane-bound organelles. So I'm gonna go ahead and define organelles. So organelles are referring to little organs that are found inside the cell. Structures within the cell. So organelles are little organs that are found inside the cell. Eukaryotes, they have organelles, they have smaller structures inside the cell. You can even see some of them on this photo. There's a lot more inside compared to this cell here. Some membrane chemistry, there's, fatty acids, non-fatty acid, lipids. I'm not gonna go too much into detail for this. If you're taking maybe chemistry class, they might go over this for you. Then the size, typical size for bacteria is 1 to 10 micrometers, same thing for archaea, but animals and plant cells, they're a lot bigger in size because they're eukaryotes again, and their size is 10 to 100 micrometers. So you wanna remember this, you wanna remember the cell size. So bacteria, they're prokaryotes. DNA is free in the cytoplasm. So cyto, you guys know what cyto means? You know what cyto means? You guys heard of cyto before? No? So cyto means cell. If a person is a cytologist, they are studying cells basically. And then plasm, it means liquid. So cytoplasm. So this is the cell, there's liquid inside, the DNA is just floating around there. So DNA is floating around and there's no nucleus. This is an animal cell. So we will go ahead and compare plant and animal cells in a second. So animal cells, they're eukaryotes. Compare this with this cell, bacteria. So prokaryotes versus eukaryotes first. So you can see there's a lot more stuff inside. And then what you can see around this nucleus, there's this membrane. And this is mitochondria. There's this membrane around it. So there's membrane almost around everything. So they have membrane bound organelles. They have little organs inside that have membrane. So what I'm gonna have you guys do, you're gonna have to go ahead and write down definitions for all these or memorize definitions for all these. And that's why I said this chapter is mostly memorization. So I'm gonna go over all these with you, but it would be a good idea to maybe do a Quizlet on the definitions for these, or maybe make flashcards for that. Maybe some of these you heard before, maybe some of these you didn't hear before. So that's animal cell. Now, if you look at animal cell here and compare it to the plant cell, can you see what's the difference? What's different between these two?
Cell wall.
Cell wall, right? So plant cells have this cell wall. It
Or
also has cell
chloroplast.
membrane, and that gives it more defined shape. So let me show you. So this is a plant cell. We already looked at this. This looks more boxy, more defined shape. There is cell wall there. And if we compare, so this is another slide you would have looked in class. I think it was the fish. And these are eukaryotic cells. So these are the egg cells. So this, you would have looked at this. And you can see this is more floppy compared to the plant cell. So plant cells more defined. This is a little bit floppy. And it's a fair game for me to put these photos on the exam, plants cell photos, or the onion cell, root cell photos, or the whitefish eggs photos, and ask you if this is eukaryotic or this is prokaryotic, or if this is onion root cell, or if this is whitefish cell. So this is just for the comparison. They look more floppy. Plant cells are more defined. They have a cell wall. So that's the cell wall. And also another thing we will talk about a little bit later today is the chloroplast. So plant cells have chloroplast. That's how they convert sunlight into food. So let's just look at the structure of the cell membrane. We have looked at this before. So there's the glycerol, and that's the hydrophilic head. Hydrophilic means they like water. They are water loving or attracted to water. Hydrophobic, phobic, water dreading or water fearing, or phobia means fear. So they repel water. They don't like water. So this part of the uh, phospholipids made of different chemicals compared to this part here. So they like water. They don't like water. So that's the structure for all the cells or the cell walls. That's the structure that you're gonna see. So each membrane, cell membrane, is gonna be formed of these phospholipid heads and hydrophobic tails. So here's a photo that you might have seen before. So I said last time, if you take these phospholipids and you put them in a test tube or in a beaker and then you shake them up, this is how they're going to arrange themselves. So there's water outside the cell. There's some water inside the cell. You're gonna notice the heads are facing the water. The tails are facing away from the water. Then there's another layer. Heads are facing outside, maybe there's water there, and then the tails are facing away from the water. What this does, creates like a sandwich. This is called phospholipid bilayer. So it's going to create a phospholipid bilayer. And then basically, this is going to create a different environment inside compared to the outside. And then if the molecules need to pass through, needs to get inside the cell, they're gonna have to go through this plasma membrane. And this plasma membrane, like I said last time, can have channels on it if it's a bigger molecule that needs to go through. Yes, so Catherine said it's like the heads are shielding the tails. So they're keeping the tails away from the water because tails don't like water. They're hydrophobic. So last time I gave you example of sodium chloride. So that would be hydrophilic. It dissolves in water. Oil would be hydrophobic. Doesn't like doesn't dissolve in water. So that'd be one example. So like I just said, if something needs to pass through this phospholipid bilayer, there can be channels there, or it can just go directly through if it's small enough. So here you see the heads, phospholipid heads, and then you see the tails here. So they're making the phospholipid bilayer. And then what you can see on this, so that's the phospholipid bilayer, you can see some proteins. So you can have transport proteins. The job of transport proteins is to take things from outside and throw them inside. So they're transporting things. Then there can be some enzymes, and we already learned last time what enzymes are. So I'm not going to have time to kind of go back to the definitions. Like I said, we're building on the material. So if you don't understand what enzymes are, you need to maybe go back and watch the recording for the last lecture. So there can be some enzymes there, and there can be some recognition proteins. Recognition proteins are same as receptor proteins. So maybe your blood sugar level is low, and your liver needs to release some glycogen. Maybe there are going to be some hormones that's going to be like, oh, tell the liver, like, knock, knock, you need to release some glucose. So that'd be an example of recognition protein. So it's going to basically start the reaction. So recognition proteins are receptor proteins. And then there are adhesion proteins. These are basically keeping things together. So they are sticking one cell to the next cell. So those are adhesion proteins. So, and that those all can be found in the phospholipid bilayer. If it's a smaller molecule, maybe it just goes through. Otherwise, it might have to use a channel. Sugar molecule is acting as a recognition protein here. So there's maybe a pattern that has to fit with this, and then the sugar molecule can pass through. Another thing you might see there is cholesterol. That's what gives us that fluidity. So I'm not going to focus too much on this. Just know that there's some cholesterol that's giving it the fluidity the phospholipid bilayer has. So this slide is showing all the membrane proteins. So I'm just going to give you definitions of these. If you maybe might want to write them down, I'm just going to kind of give you what each one of these are. So we have intercellular joinings. So those are type of adhesion protein. It's just sticking things together. So intercellular joinings are going to stick two cells together. Then we have enzymatic activity. Last time when I showed you an enzyme, I said a substrate can come in, attach to the enzyme, and enzyme is going to break it down into products. So that's just going to break down maybe food. That'd be one example. And that's enzymatic activity. Transport protein, I already told you they're going to grab stuff from one side and shoot it to the other side, either from inside to outside or outside to inside. So they're going to transport things. Here you will notice it's saying active or passive. Active means basically it's going to need energy to do the job. Passive, it doesn't require any energy. And we learned ATP. You guys remember what ATP meant? ATP is the energy here. It's a energy carrier molecule. You guys remember what ATP stands for?
Adenosine triphosphate.
Yes, so make sure you guys know adenosine triphosphate. Each time that phosphate bond breaks, it's going to release energy, and that's next chapter. And then recognition protein. So how do you know if you're blood type A and you get blood type B, what's going to happen? So
Your body's going to reject it.
Your body's going to reject it. So there are some recognition proteins found in your blood cells that your body recognizes. If somebody gets organ transplant and their body doesn't accept it, they reject it, that means there's a different recognition protein found on there. So those are the recognition proteins. So the blood is going to start clumping if you get a different kind of blood. Your body's going to try to attack it. They're gonna think it's something foreign that's trying to hurt you. And then there's anchorage or attachment. These are basically holding it in place. So this is showing that it's holding this whole thing in place. So it's otherwise your body will fall apart. So it's holding it in place, anchorage or attachment. Next one is signal transduction. This is similar to recognition protein. Basically, this has to attach here, maybe for this channel to work. So a substrate maybe has to attach here for this channel to work. So it's going to send a signal for this channel to open maybe. So same thing as a recognition protein.